word. And you might have heard the Dictionary of Fungi by Ainsworth. In 1991, he reported that there are about 1.5 million species of fungi which are occurring around the globe, which have not been identified only at that time. 70 to 80,000 fungi were identified when I am talking of 1991. And now the estimates on the basis of what we call as the morphological characteristic features, they are the basis, they are the backbone. And then we talk of the molecular DNA techniques. What has been estimated now, there are 12 million species which are there in nature, occurring in nature in various habitats. And out of these 12 million species, we have identified nearly 150,000 species as the latest information in fungal biodiversity is. And how much time it will take? That will take another thousands of years if we have to identify the 12 million species which are prevalent in nature. These are the estimates. There's a recent paper in 2022. There's a recent paper I have gone through just one day advance when I was coming here for attending the conference. 12 million species estimated and 1.5 lakh species which have been identified. Now when we started, as we say, taxonomy is difficult, but how to start this taxonomic, basic taxonomy? When we talk of the what we call as the Necraceous or Zygomyces fungi is concerned, the methodology or the characteristic feature, they are different. When we talk of the Ascomyces, they are different. When we talk of the Basidiomyces, they are different. You know, our earlier lecture says most of the mushrooms which have been taken by Professor Saab from Professor Lakhanpal. If Professor Lakhanpal would not have identified those mushrooms, where would have been our nanotechnology? Nanotechnology comes later, biotechnology comes later, basic comes the taxonomic studies in mycology, and that's the term which we are using as the fungal biodiversity nowadays. Fungal diversity means variability of fungi in various habitats. Variability means total species of fungi. Of course, in addition to that, if we go broadly, what the biodiversity is concerned, not only the identification, their applications too, that comes under the umbrella of when we talk of the biodiversity or fungal biodiversity. So my first lecture comes with that. I started work for my PhD in 1974, and my problem at that time was taxonomical, ecological and physiological studies on soil microflora decomposing the plant waste or plant litter. That was in 74, and I got my degree in 79. What I did? I studied the fungal succession. That is the colonization of the substrate at different stages of decomposition. As you know, when we start up the decomposition of any substrate, we are talking of now how we can utilize the fungi for biodegradation. We are facing lot of problem in Delhi as the pollution is concerned. Still, solution has not come. How we can hasten the activity of those microbes or fungi, bacteria, which are occurring on that very substrate. The only solution is selection of those potential microorganisms which can be utilized for their degradation. That is the only solution. And I started at that time. It was not only identified what we call thousands of species were isolated. Identified myself, we were not having the technology. These samples or along with the cultures plus the substrate were sent to at that time name was Commonwealth Mycological Institute. Now it is International Mycological Institute. And CBS Netherlands Center collected, isolated, identified, then centers culture sent for the verification of the identification there, then you can publish the papers. Otherwise, publication was not difficult unless otherwise the culture is identification is authenticated. This was the procedure followed at that time. We were sending it and we were identifying there. Verification was very essential. But that was continuously sitting in the laboratory for 18 to 20 hours I was sleeping there in the lab itself, not going. I belonged to Kurukshetra, I remained in the city, but not going to the residence in the night, sitting in the lab itself, sleeping in the lab itself during those days. Identification was so difficult. Along with the identification, I did what physiological studies mean. Their cellulitic activity, their lignolytic activities, their pectolytic activities, all the enzymes which are secreted by the microbes, they were being studied by following the standard technology, whether it was by the viscosity method. If you want to estimate the CX activity by the viscosity method, C1 activity by the filter paper, cotton, and the cellulose powder utilization, then the pectolytic enzyme by applying pectin to the substrate, and then the lignolytic activity on the substrate by applying tannic-like substances. These were the methodology, and I studied about, I think, more than 100 species of fungi for their activity. And then after that activity, I went for their what we call as the role in vitro decomposition, taking the litter, inoculating pure fungus and mixture consortia for seeing how whether they can be utilized that for their decomposition or not. Then what I did, saw their correlation. What the enzyme they are producing, cellulosic enzyme, pectolytic, lignolytic enzyme, what they role have in colonization and decomposition. What all I was seeing during my PhD time when I did PhD under the guidance of Professor R.S. Maran. But for the help in the taxonomic identification, I went to Professor K.G. Mukherjee's lab in Delhi University, Botany Department, Delhi University at that time, because he was the known mycologist at that time. My slide, please. Now, when we talk of the decomposition, I will start with this. When we talk of the decomposition, two patterns of colonizers were known at that time. One pattern was the pattern suggested by the Garrett, 1960, that is the American, and another was by Hudson, British, 1968. That if a land substrate is going to be colonized, what are the organisms, what are the fungi which are grown, and what is the pattern of the cell? But we know, Garrett suggested on the basis of their utilization, whether they are the first coming sugar fungi, they are followed by the cellulosic fungi, and they are followed directly because lignin is the last to be decomposed by the lignolytic fungi. Sugar fungi followed by the cellulosic fungi, and that's it, followed by the lignolytic fungi. And when we talk of the sugar fungi, as you know, they are the nucleaceous fungi belonging to the nuclease of phycomycin or zygomycin, what and everything. Then when we talk of the ascomycin and epiromycin, they are the cellulosic fungi. And when we talk of the what we call as the lignin decomposing fungi, you know, they are the pseudomycin fungi. But not only that, these are the mushrooms which have the ability to utilize lignin. These what we call the fungi. So the fungi which belong to the Deuteromycetes, they have the ability to utilize lignin, and they are have been seen when we talk of the what I took three substrate: Desmostachia bipinnata, grass litter, Chenopodium album, herbaceum, and mixture of grasses for studies. And when I saw the decomposition of the samples were decomposed at the soil surface, below the soil surface, and in the aerial at three different positions as the decomposition pattern was seen at that time. My first slide. These are research areas. Of course, I started from the basic taxonomical studies, or when we talk of the diversical studies, then I went to their application. That is the how they can be used for the application of the mushrooms is Agaricus bisporus is concerned. As their ability is concerned, it can be utilized for their stimulation because thermophilic fungi are involved. I work on the thermophilic fungi. Then my other area was activity of herbal plants. That activity, that is the food spoilage, and then the lastly I worked in the area of mycoherbicides. First slide, please. This is my PhD work. Different. That is the succession of fungi, production of the extracellular enzymes, cellulitic, pactolytic, linolytic, and their potential exploitation in the decomposition as the individual role is concerned. These are the pattern, Garrett pattern and the Hudson pattern. Oh, Nira. This is the discuss because time is very less. These are the papers which have been published in this area, whether it is the taxonomy, whether it is the pattern of colonization and the role in decomposition. What is the important in this is चलता चल नहीं रहा इधर आओ भैया अपना अच्छा अच्छा ठीक अच्छा चलिए आप नेक्स्ट बोलिए नेक्स्ट how much I have identified 42 fungi and all these I have identified 100 of fungi but out of these 42 have been reported to be new record from the country substate as well as from the globe. Next, these are cases of all those fungi which have been published. Next. Where is the thermophilic fungi? I worked on thermophilic fungi from two different climate, temperate climate as well as tropical climate. Saw the comparison. What is the difference between the two different type of climates? What was the interesting? It was not only that the tropical climate is having the higher temperature and you will find the thermophilic fungi more. It was not. It was reverse to that. Temperate climate was Shimla, Solan was having more thermophilic fungi and it was seemed to be that their if we talk of the origin, they are there, then they come to the what we call as the tropical climate in the plains is concerned. It was a very interesting study. Next, then what is another basis? I studied again on the enzymatic work, thermophilic fungi, and then their utilization of these thermophilic fungi, how we can enhance the agaricus bisporous production in the laboratory. Next, then what was the interesting point here is we wrote a monograph on thermophilic fungi. 42 species of fungi which were known at that time. They were categorized, identified, and that is very, very useful for the identification of all the thermophilic fungi which have been known till today from the world. These are information on the thermophilic and thermotolerant fungi. Next. Here is the spoilage, various substrates, especially your pickles, carrots, they were studied. My latest publication is there on the juices, that is the spoilage of juices by fungi and bacteria. So many fungi have been reported, and you know when we talk of the fungi, if we talk of the raw juices, they are having the molds, and some of these molds are, as you know, the toxin producers, Aspergilli, Penicillii, they are the toxin producers, and these toxins, you know what these toxins, they are carcinogenic one, and we studied what? Preservative, plant preservative were used that we can, how we can preserve these juices on the basis of the plant extract. Next, please. Next. These are bacterial and fungal isolated from the pickles. Next. These are activity associated with this. Next. These are publications. Now, I come to these is we studied on 3 human pathogenic fungi. One is the dental caries causing pathogen, another is ear pathogen, another causing candidiasis, your oral and vaginal candidiasis. These were isolated from Delhi as well as Kurukshetra health clinics. They were, after their isolation, they were identified in the laboratory. When they were identified in the laboratory, then they were evaluated with different plant products, that how we can control with the help of these plant products. Hundreds of plants, so hundreds of medicinal plants were evaluated for their activity. When we talk of the extract, aquatic and organic extracts were used. They were evaluated by in the petit plate method.
after finding the good organism or plants which were inhibiting the growth of these plants, they were isolated for the chemical compounds. What are the chemical compounds which shows good activity, and they were identified up to their identification level. Next, please. Next, 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 next. These are the ear, that was the dental caries pathogen. This is against the ear pathogens. Next, next, next. And these were the two compounds which have been isolated. One later, another from the Jugulus regions which have been identified, and these two compounds have shown the ability to control your ear pathogens, and there is possibility that they can be used for the evaluation of as ear pathogens is concerned. Ear control pathogens as the compounds for their products are concerned. Next, these are two compounds which have been found to be the best. They have been found to be positive. They have found the ability to control your ear pathogen, that is the fungal pathogens, bacterial pathogens, both gram positive and negative, means they are wide spectrum in activity. Next. Next. These are applications in this area of research. Now, in addition to that, what was done in our laboratory, that we evaluated the plants for their antimicrobial activity against these pathogens. Then we interacted with the chemistry department and the pharmaceutical department in Kurukshetra University itself. What were they doing? They were synthesizing the compounds, and these synthesizing compounds were analyzed by us for their antimicrobial activity, and over 50-60 papers, high impact journals have been published in coordination with the chemistry and pharmaceutical departments. Next. Then we come to the most important area of research. As we know, the emphasis is on the development of bioherbicides or mycoherbicides. That is the using of weeds by applying the microbes. And when we talk of the microbes, of course, three groups of organisms are being used for controlling these weeds. They belong to bacteria, they belong to viruses, they belong to fungi. Till, as the literature says, latestly 22 products are available around the globe which are being used for controlling the various weeds. And out of these 22 products, there is only one fungal product and one or two virus product. Rest all belong to the fungi, all fungal pathogens. Now, when we talk of the terms, if you see the literature, you will find bioherbicides and mycoherbicides have been interchangeably used because most of the products are based on the fungi, not on the bacteria and viruses. I also worked on this as the biological control of three important weeds are concerned. One is water hyacinth, another is trianthema, and third is your parthenium. These three. I was having three products, what we call 4 projects from different funding agencies, 2 from the UGC, 1 from the Department of Environment, and 1 from the ODI UK. Terrestrial weeds from the UGC. For the aquatic weed, it was from the Department of Environment, and for the parthenium, it was from the UK. And I was the first and the only scientist which brought direct funding grant from the UK to our country, our university. That was the first project which was sanctioned in 1996, and that project ran for 4 years, 1996 to 2000. Now, next. This is a 126 weeds. Hundreds of pathogens were isolated from 26 weeds, but out of these 26 weeds, I selected only 3 as I've already mentioned you. Next. Next. These are the 19 fungal pathogens which have been reported for the first time on this very wheat from our country, as some of these which have been reported for the first time from the world. This is the list of the 19 fungal pathogens which have been identified on water hyacinth, which are new. Next. So here is the 3 pathogens. Out of these, 7 fungal pathogens have been tested for their potential, whether they can be used for controlling the water hyacinth or not. Out of these 7 fungal pathogens, you know, it is very simple. I'm explaining in one word. But when we talk of the evaluation of a one product, it's a very lengthy process. It takes 5 to 7 years. If you start with one fungal pathogens, if you have to reach for controlling it, minimum 5 to 7 years are desired. Because if we have to start with the work on a mycoherbicide, first what we have to do, we will have to go to the field for selecting the virulent pathogens. After the selection of the virulent pathogens, we have brought those specimens to the laboratory. Then we have to go for the isolation. After the isolation, we have to go for the identification of the pathogen. After the identification of the pathogen, we have to go for testing of the pathogenicity and cost postulate. When pathogenicity and cost postulates are proved, then we have to go for the evaluation. And when we have to go the evaluation, if it is found, it is controlling in vitro conditions, then we will have to go to the vivo condition. If it is controlling in the vivo condition, then we will have to go for testing its toxicity. Then after the toxicity, the most important parameter is we have to test for its host specificity, whether it is host specific or not. Because a fungal pathogen, if it has to be used as a mycoherbicide, cannot be used as a mycoherbicide unless otherwise it is found to be a host specific. And this is what we call as the scheme if we have to start for the starting of any organism to use as a biocontrol agent or to be used as a mycoherbicides. So these are the 3 fungal pathogens which have shown the activity, which have shown the activity which can be exploited for biocontrolling it by use of the fungal pathogen. Because what is going on, if we talk about country, till now no herbicide is available. Of course, the work was the biological control of the weed in our country started in 1970 itself. In 1970, in the south, water hyacinth, there was a paper which was published. Alternaria shorni was reported, published for the first time. That was the new fungus which was reported from South India by Nagaraj and Punappa in 1969-70 for the first time. Before even what we call as the Americans are concerned, they reported later on, they started the control of weeds later on, and the fungus was reported first in our country. But the work was carried over for its as the evaluation is concerned. The work remained as such. Publication was there. Paper was published in Transaction British Mycological Society in 1970, but it was not pursued further. Now, what I have found here, as I was telling you, I have found 3 pathogens. Alternaria alternata is here, Alternaria shorni is there, and Cercospora rhodmani is there. These 3 pathogens have been found to be the best, which have shown the efficacy, which have shown the potential to be developed as a mycoherbicide. And I am recommending these. I have just sent one paper. I have just for Kavka, they wanted me to have a paper. I have sent that paper. All the results of the details have been given of these 3 pathogens there. And I want a formulation to be made of all these 3 fungal pathogens, because these fungal pathogens have been found to be synergistic. They have not been found to be antagonistic. They have been found to be synergistic. And another important feature is what I have seen here, that we have tested the Philosphaera fungi which are coming on the water hyacinth, and they have been screened against all these pathogens, and they have not shown, any of the colonizer has not shown any antagonistic activity. So there is no restriction, no reduction will happen if we go for the formulation of these fungi as one preparation, and if we go what we call as the commercial application of this for biocontrolling, it is. Because I have been made recently member of the research advisory committee of the ICR Center, Jabalpur, WIT Center. So I will be going for the meeting in the next February meeting. First meeting will be held in February, and I will be going because they are using this neocatina ashorni for biological control. Different ponds in the area of Jabalpur. And they are successful in controlling with that neocatina ashorni. But what one problem they are facing is neocatina ashorni takes lot of time in controlling that very weed is concerned. And there are publications, a recent publication is there, and publications are there. If you use a fungal pathogen with the neocatina ashorni, because neocatina ashorni will make a passage for the anti, that will make a wound, make a passage for the anti of the pathogen, and that will boost the potential of the fungal pathogen. And if they use fungal pathogen with the neocatina ashorni, that problem, the duration, that problem can be resolved if we use fungal with the insect
Sir,
as biocontrol agents is concerned.
Can we wind up in another 2 minutes? We are already 25 minutes.
Next. So these are next. Chaliye, slide. Next, please. These is on the water essence. Next. These are publications. These are the publications of water essence alone. Next. Here is the control of Parthenium. I was having a project from UK on the control. Only 2 pathogens have been found to be efficacious for controlling this. One is Cercospora partheniphylla and another is Alternaria gini in our country. Next. There is another, this is the formulation which have been made by me on this as the horse purslane. Trianthema is concerned. Gabarenth. I have given this name. This formulation is based on the fungus Gibago-trenthmi. Trianthema is the host, Gibago-trenthmi is the fungus, and this fungus is a very interesting and unique fungus. And what is the special? This fungus was reported first of all from the United States of America on this very host. After that, no work was carried out. When we started work at Kurukshetra, we isolated this fungus from Kurukshetra and Haryana, different parts of the Haryana. We evaluated this, and we saw all the features for the evaluation to the what we call as the field trials as concerned. Work was carried over for 15 years or so, and after the 15 years, this formulation was made. Based on the next. Here the next. These are the six fungal pathogens which have been reported on this very host from Kurukshetra itself. Next. These are the publication on this Trianthema gibago and other pathogens. Next. These are publications on this Trianthema. Next. Now, this is the last slide I was telling you as the fungal diversity is concerned. You see, this is the latest report. I was telling you, Gautam et al. 2022. This is the recent paper which has come on the diversity, fungal diversity is concerned. All these have been given, and what is estimated now, I have told you that 12 million species, the estimates say there are 12 million species which are occurring in nature on different spreads. And if we start as the rate of identification is concerned, if we have to identify these estimated species which are occurring in the nature, that will take 1,000 years so, which has been suggested by these authors. This recent paper says. This is on the fungal diversity. Next. Now here, this is the list of the 10 books. There are 3 edited books and 9 authored books. 9 authored, 3 edited, and there are 2 volumes which have been written by me, authored by me, edited by me. Next. Now here is the 2 books have been released today in the morning session. This is the another book which I wrote in 1991. First edition of this book, Experiment. Some of you will be aware of this book. It was written in 1991. And the 6th edition of this book is likely to be released in January itself. And this is the photograph of that 6th edition. It has not been released. It will be released in January 2023. Next. So this is if we have to see, nature is there. We are getting so many things from the nature. But there was another paper that there are over 50 fungal formulations are there which have been commercialized till today. This was a recent paper. All these details have been given in that. But story doesn't end here. There are not only 50, as nanotechnology says, there are so many products which are to be explored from the fungi. Now we have to work again on the fungal diversity. That is the identification to their applications if we have to do it. And this is for the budding scientists that they have to do more. Lot of opportunities are there, but what is there? Hard work, determination is desired if we want to achieve that objective. Next. Here is, I'm very thankful to my scholar, Dr. Vikas, who helped me for preparing all this as the presentation is concerned. I'm very grateful to him for that this presentation is concerned. Thank you.
So we thank you, Professor Aneja ji. He was talking he gave this lecture from the vast knowledge, practical experience. We congratulate you, sir, for this much deserved Lifetime Achievement Award. I would like to invite Dr. N. Madhivanan for introducing the next lecture and to honor Professor Aneja. I invite Professor Lakhan Palji to present the certificate of appreciation to Dr. Aneja. So next in line, we have Professor C.V. Subramanian Memorial Award Lecture. I would invite Professor N. Madhivanan ji to introduce this lecture, the importance of this lecture, to be delivered later by Professor C. Manoharacharya ji.
Thank you, Chairman, for inviting me to read out the biography of the eminent mycologist, who is the founder of Mycology Society of India, none other than Professor C.V. Subramanian. Most of the senior mycologists know very well about the contribution of Professor C.V. Subramanian. This is for the benefit of the young mycologist and all those students participant. I would like to read the salient contribution of Professor C.V. Subramanian. Though the detail is very lengthy because of want of time, I wanted to cut short. Very important points I wanted to read out quickly. Late Professor C.V. Subramanian, affectionately referred to as CVS, was an internationally renowned and most distinguished mycologist of our country. Professor Subramanian was a complete mycologist